Antony van Leeuwenhoek, 1632 to 1723, stands as one of the most important figures in the history of science, renowned primarily for his pioneering work in microscopy. While his name may not immediately come to mind alongside figures like Galileo or Newton, Leeuwenhoek's contributions fundamentally reshaped the landscape of scientific discovery. His keen observation and meticulous attention to detail allowed him to observe the invisible world of microorganisms, laying the foundations for the field of microbiology. This chapter aims to introduce Leeuwenhoek's life, contributions, and the significance of his work, which altered our understanding of the natural world in profound ways. Born in Delft, the Netherlands, in 1632, Leeuwenhoek came from modest beginnings. His early life was not marked by formal academic training. Rather, he was a self-taught man, driven by a curiosity about the world that led him to become one of the first people to observe microorganisms in their natural environment. His professional life was centered around the trade of cloth, where he worked as a merchant and draper. However, it was his passion for lens making and his experiments with microscopes that eventually brought him lasting fame. What distinguishes Leeuwenhoek from many of his contemporaries is not only his observations, but the tools he crafted to make those observations possible. His microscopes were not merely instruments. They were windows to an entirely new world. Leeuwenhoek's work was groundbreaking in several key areas, particularly in the realm of microscopy. His development of single-lens microscopes, which magnified objects up to 200 times, was unmatched by the technology available during his time. Through these microscopes, he was the first to observe and document the existence of bacteria, spermatozoa, and red blood cells. This revolutionized biology, offering evidence of a microscopic world that was previously unimagined. While scientists before him, such as Robert Hooke, had glimpsed tiny structures through the microscope, Leeuwenhoek's attention to fine detail and his insistence on personal observation and verification set him apart. What truly sets Leeuwenhoek apart from other scientists of his era is his emphasis on empirical observation. He did not rely on the theoretical explanations that were popular at the time. Rather, he meticulously recorded his observations and shared them with the scientific community through letters. In fact, much of what we know about Leeuwenhoek comes from his extensive correspondence with the Royal Society of London. These letters, filled with vivid descriptions of his observations, were key in disseminating his findings and establishing his reputation as a scientist. Leeuwenhoek's method of sharing his discoveries also highlights another important aspect of his character: his reluctance to share the secrets of his craft. Despite his correspondence with other scientists, Leeuwenhoek was famously secretive about his methods for crafting lenses and constructing his microscopes. This secrecy, while frustrating to some of his peers, ensured that his contributions remained uniquely his. In a way, this guardedness about his tools also contributed to his legacy, as it allowed for the development of techniques that would inspire future generations of scientists to innovate and experiment in their own right. Although Leeuwenhoek's discoveries were monumental, it is important to acknowledge that his work did not initially receive the widespread acclaim that it deserved. His discoveries were so far ahead of their time that many of his contemporaries did not fully understand or appreciate the significance of his findings. It was only through the dissemination of his letters to the Royal Society and other learned institutions that his ideas began to gain recognition. His work, however, remains somewhat peripheral to the broader scientific discourse of the time, which was dominated by figures like Isaac Newton and René Descartes. The significance of Leeuwenhoek's discoveries became clearer with the passage of time. The existence of microorganisms, which Leeuwenhoek described in his detailed letters, had profound implications for various fields of science, particularly medicine and biology. His observations set the stage for the later development of germ theory, which would be articulated more fully by scientists like Louis Pasteur and Robert Koch in the 19th century. Leeuwenhoek's work also laid the foundation for the field of microbiology, which would become essential to our understanding of disease, ecology, and the vast diversity of life that exists at the microscopic level. It is crucial to note that Leeuwenhoek's approach to science was unique in its insistence on direct observation and evidence. While many of his contemporaries were content to work with theoretical models and abstract principles, Leeuwenhoek believed that only through direct observation could true knowledge be gained. His insistence on this empirical approach marked him as a forerunner of modern scientific methods, which continue to emphasize observation, experimentation, and evidence-based conclusions. Today, the legacy of Antony van Leeuwenhoek is celebrated in the field of microbiology and beyond. His ability to unlock the secrets of the microscopic world and his dedication to empirical research are central to the development of science as we know it. The very techniques he used in his microscopic investigations, careful observation, recording, and sharing of findings, remain foundational to modern scientific practice. Leeuwenhoek's work serves as a reminder of the importance of curiosity, attention to detail, and the willingness to challenge conventional wisdom in the pursuit of knowledge. Antony van Leeuwenhoek's journey to becoming one of history's most celebrated scientists began in the small Dutch city of Delft. Born on October 24, 1632, into a family of modest means, his early years were far removed from the grandeur of the intellectual elite. His father, Philips Antinous, was a basket maker, and his mother, Margaretha, was a homemaker. Antony was their second child, and his early life was characterized by the kind of simplicity typical of many children in 17th century Dutch society. Yet, even in this unassuming environment, his inquisitive nature began to take shape, setting him on a path that would lead to extraordinary scientific discoveries. Leeuwenhoek's formal education was brief and unspectacular, a stark contrast to the rigorous academic paths followed by many of his contemporaries. At the age of 5, he enrolled in the Latin School of Delft, a school where he learned the basics of reading, writing, and arithmetic. However, his education was cut short when, at the age of 16, he lost his father. With his father's death, Leeuwenhoek became responsible for supporting his family, and he transitioned into the workforce, leaving behind formal schooling. Despite this, his natural curiosity and self-directed learning would remain a central part of his life. After leaving school, Leeuwenhoek took an apprenticeship with a merchant in Amsterdam. This was not only a step towards securing financial stability, but also an opportunity for him to explore the world beyond Delft. While working as an apprentice, Leeuwenhoek began to develop a deep interest in the craft of lens making. His exposure to a wide variety of objects during his apprenticeship sparked an interest in the precise methods of magnification and observation. As he immersed himself in the world of trade and lenses, he developed a growing fascination with the possibilities of seeing things that were beyond the range of the naked eye. Leeuwenhoek's early training in the world of commerce proved useful in shaping his future endeavors. In 1654, at the age of 22, he returned to Delft and established himself as a merchant and draper, dealing primarily with cloth. Despite his career in trade, Leeuwenhoek's mind remained restless, forever drawn to the details of the world around him. It was in this period of his life that he began experimenting with lenses, building on his skills to design and craft his own microscopes. His success as a merchant provided him with the financial means to pursue these interests more seriously, but it was his intellectual curiosity that truly drove him. Though Leeuwenhoek had no formal scientific education, his keen observational skills and persistence led him to experiment on his own. He began by crafting simple microscopes, single-lens devices with magnification powers far superior to anything available at the time. The tools Leeuwenhoek designed were unlike any that had been previously created, and their effectiveness was a direct result of his hands-on approach to understanding optics. He spent years refining his craft, producing microscopes with magnification powers that far exceeded anything the scientific community had seen before. It was through these microscopes that Leeuwenhoek would come to observe the previously hidden world of the microscopic, making discoveries that would forever alter the course of science. In many ways, Leeuwenhoek's self-education was a reflection of his intellectual independence. Unlike many scientists who were trained in universities or scientific academies, Leeuwenhoek learned through personal experience, trial, and error. His work was not shaped by a specific scientific institution, but was instead an individual pursuit fueled by a lifelong passion for discovery. This self-directed learning allowed him to develop a unique approach to science, one that prioritized hands-on experimentation and direct observation over theoretical concepts. Leeuwenhoek's intellectual independence and commitment to personal discovery will be defining characteristics of his scientific career. Despite his lack of formal training, Leeuwenhoek's curiosity led him to make observations in areas that most of his peers overlooked. One such area of interest was the study of human anatomy. He became particularly fascinated with the structure of the human body, especially the vascular system and the microscopic components of bodily fluids. Using his microscopes, he was able to examine human and animal tissues with a level of precision that had never been achieved before. His exploration of spermatozoa, for example, was groundbreaking, revealing the existence of tiny living organisms within human semen, a discovery that would forever change the understanding of reproduction. Throughout his life, Leeuwenhoek remained deeply interested in the natural world, and he spent considerable time observing not just biological specimens, but also environmental objects. He would examine everything from pond water to the surface of a grain of salt, noting the minute details and strange new organisms that appeared under his lenses. His meticulous notes, written in the form of letters to fellow scientists, were filled with descriptions of these creatures, which he often referred to as animalcules. These letters would eventually form the foundation of his reputation, and they were pivotal in spreading his ideas throughout Europe. Although Leeuwenhoek's education was informal, his scientific method was anything but haphazard. He was deeply committed to documenting his observations with extraordinary precision, often recording his findings in detailed letters to members of the Royal Society of London. These letters were filled with careful descriptions and intricate drawings of the things he saw through his microscopes. His ability to convey the results of his experiments with clarity and accuracy earned him respect and recognition among the scientific community, despite his lack of formal training. His correspondence with leading scientists in England allowed him to present his discoveries to a wider audience, helping to establish him as one of the foremost pioneers in the field of microscopy. Leeuwenhoek's lack of formal education did not hinder his ability to make profound contributions to science. In fact, it could be argued that his absence from the structured, academic world of science allowed him to approach his observations with a fresh perspective, free from the constraints of established dogma. His discoveries were not limited by conventional thinking, and it was his open-mindedness, combined with his practical skills, that enabled him to make discoveries that were years ahead of their time. Antonie van Leeuwenhoek's path to scientific greatness was shaped not only by his natural curiosity, but also by his career as a merchant and draper. Born into a family with modest means, Leeuwenhoek's early adult years were marked by the need to establish himself financially. In 1654, at the age of 22, he returned to Delft, the city of his birth, and started his own business as a cloth merchant. This career allowed him to rise in social status and gain financial stability. While his business dealings brought him practical success, it was his growing interest in optics and lenses that would ultimately elevate him to the annals of scientific history. Leeuwenhoek's decision to settle in Delft was pivotal. Delft, a thriving city in the Dutch Republic during the 17th century, was a hub of intellectual activity, though Leeuwenhoek himself did not immediately immerse himself in the formal academic world. Rather, he began his life as a merchant, selling cloth to customers and managing his business affairs. His daily life was not unlike that of any other cloth merchant, but there was a critical difference. Leeuwenhoek's exceptional attention to detail and his keen interest in precision set him apart. Even as he focused on his business, he would often find himself examining the small details of the world around him, a tendency that would soon lead him into the world of lens making. In his early years as a merchant, Leeuwenhoek developed an increasing fascination with optics. His interest in lenses and magnification began as a hobby. He would experiment with the design and crafting of lenses, a skill he had learned in his earlier years when he was exposed to the world of glassmaking and optics during his apprenticeship in Amsterdam. However, it was in Delft where his passion for crafting lenses truly flourished. Leeuwenhoek's desire to understand the smallest components of the world around him led him to devote hours to perfecting the lenses that would enable him to do so. This obsession with precision in the art of lens making became the cornerstone of his future scientific endeavors. Leeuwenhoek's ability to craft such lenses was groundbreaking, as his microscopes were among the most advanced of his time. Unlike the compound microscopes that were being developed by his contemporaries, which utilized multiple lenses to achieve magnification, Leeuwenhoek's microscopes used a single, meticulously ground lens. This innovation allowed his instruments to achieve a level of magnification that was far superior to anything else available at the time. His lenses, though relatively simple by modern standards, allowed him to see minute details in objects that had previously been hidden to the naked eye. At first, Leeuwenhoek's microscopes were primarily used for his own personal curiosity as he began to explore the microscopic world around him. He observed everything from the fibers of cloth to the structures of insect eyes, gradually refining his understanding of the powerful tool he had created. It was not long, however, before he began using his microscopes to investigate even more complex subjects, including human and animal tissues, water samples, and even the blood of various creatures. His success in making detailed observations with these instruments gave him the motivation to delve deeper into the world of microscopy. His experiments were not merely the product of idle curiosity. Leeuwenhoek applied a systematic approach to his observations, documenting his findings with remarkable accuracy. He would meticulously draw what he saw through his microscopes, and he would record the results of his experiments in detailed notes. These drawings and descriptions, along with the magnified observations he made, were later shared with other scientists, particularly through his letters to the Royal Society of London. Though Leeuwenhoek did not receive formal training in science, his hands-on experimentation and his careful record-keeping gave him a level of credibility in the scientific community. While Leeuwenhoek was growing in reputation as a skilled craftsman and inventor of a microscope, his role as a merchant was still central to his daily life. He continued to manage his cloth business and made a comfortable living from his work. His dual roles as both a businessman and a budding scientist provided him with the financial means to continue his lens-making experiments. Yet it was his scientific discoveries that would eventually eclipse his success as a merchant. His ability to balance both a successful career in trade and a passion for microscopy is a testament to his discipline and intellectual ambition. As he honed his craft, Leeuwenhoek began to draw attention from some of the most prominent scientific minds in Europe. His microscopes and the observations he made through them were unlike anything other scientists had seen before. His letters to the Royal Society in London were the primary means by which his discoveries were shared, and through these correspondences, he was able to engage with other intellectuals. His reputation as a skilled microscopist began to grow, and he became known as the father of microbiology in certain scientific circles. Despite the growing recognition of his scientific contributions, Leeuwenhoek remained resolutely independent. He did not see himself as part of the academic or intellectual elite, and he continued to run his cloth business. He did not feel the need to align himself with any institution or formal academic structure, preferring instead to work alone. This was in sharp contrast to other scientists of the time, who were often part of established academic networks. Leeuwenhoek's autonomy allowed him to follow his instincts and continue to explore new avenues of discovery without the constraints of institutional pressure. In his later years, as his fame as a scientist grew, Leeuwenhoek continued to work from his modest workshop in Delft, where he would often be found crafting microscopes or conducting experiments. He continued to engage with the Royal Society, and his letters continued to be a vital source of information about the microscopic world. His life as a merchant never completely faded, as he remained financially independent, but it was clear that his true passion lay in the pursuit of scientific knowledge. In many ways, Leeuwenhoek's career as a merchant and his work as a lens maker laid the foundation for his greatest scientific achievements, demonstrating that a career in trade need not be mutually exclusive with intellectual achievement. The moment Antony van Leeuwenhoek turned his lenses upon the world, a new era of scientific discovery was born. Though his early microscopes were crafted for the mere purpose of magnification, Leeuwenhoek's curiosity led him to something far more extraordinary: the discovery of microscopic life. What began as a personal exploration soon transformed into groundbreaking observations that would forever change our understanding of biology and the natural world. Through his single-lens microscopes, Leeuwenhoek was able to peer into an invisible realm, encountering organisms so small that they defied previous comprehension. These discoveries laid the foundation for the burgeoning field of microbiology and introduced the world to the existence of life, forms unseen by the naked eye. In the early 1670s, Leeuwenhoek began experimenting with various samples under his microscopes, fascinated by the details of everyday objects that seemed mundane but were, in fact, teeming with life. One of his first subjects was a drop of water from a pond. Upon viewing the sample through his microscope, he was astonished to find that the water was alive with tiny organisms, which he described as animalcules. This observation marked the first time that a scientist had documented microorganisms in such detail. Leeuwenhoek's meticulous observations allowed him to identify these tiny creatures, noting their movements, behavior, and distinct structures. He was astonished by the diversity of life in the smallest of environments, and it was this revelation that would set the stage for his subsequent discoveries. Leeuwenhoek's fascination with animalcules extended beyond pond water. In the years that followed, he turned his attention to other sources of microscopic life. He examined the human mouth, where he found an array of bacteria, and was one of the first to describe the presence of microorganisms in human saliva. His observations of these minute creatures provided evidence that life was not restricted to the larger, visible world, but was instead omnipresent, existing in even the most unexpected places. He also began to explore the feces of animals, the blood of various creatures, and the fluids from the human body, all of which revealed the same startling truth: the existence of a world of life forms that had previously been invisible. One of Leeuwenhoek's most profound discoveries was the observation of spermatozoa, or sperm cells. By examining human semen, he was able to identify these cells for the first time, observing their movement and suggesting that they played a role in the process of reproduction. While the full significance of this discovery would not be understood for centuries, Leeuwenhoek's observations marked a crucial step in the understanding of human biology. The idea that there was a microscopic world within the human body was revolutionary at the time and underscored the vastness of the unseen universe that Leeuwenhoek was uncovering. Leeuwenhoek's observations extended beyond the human body. He also looked at a variety of other natural samples, from the wings of insects to the texture of cloth. With each new object, his microscopes revealed new and previously unknown details. He examined the structure of hair, the internal structures of the eyes of insects, and the tiny scales of fish. His attention to detail was extraordinary, and his ability to see these fine features, which had been hidden from previous generations of scientists, was an achievement that set him apart. Every discovery seemed to open up new avenues of exploration, and Leeuwenhoek's growing catalog of observations made him one of the most important naturalists of his time. What made Leeuwenhoek's discoveries particularly remarkable was not just his ability to see these creatures, but his skill in accurately documenting them. Unlike many early scientists who would rely on illustrations or verbal descriptions of their findings, Leeuwenhoek's microscopes allowed him to make precise observations and then record them in detail. He would often sketch what he saw through his lenses, providing evidence of his findings and contributing to a growing body of work. His ability to describe the minute characteristics of microorganisms, often in vivid detail, made his work indispensable to the scientific community. Despite the remarkable nature of his discoveries, Leeuwenhoek faced skepticism from some quarters of the scientific community. His findings were so far ahead of their time that many could not comprehend the full significance of his work. The idea that there were creatures living in water, in the human body, and in other natural environments was revolutionary, and some viewed his discoveries with doubt. To address this skepticism, Leeuwenhoek sent his detailed letters and observations to the Royal Society in London, where they were published and presented to other scientists. While some initially questioned his claims, the clarity and consistency of his observations gradually won over even the most skeptical members of the scientific establishment. One of Leeuwenhoek's most important contributions to science was his approach to evidence. Rather than relying on theoretical models or conjecture, he placed great emphasis on direct observation and empirical data. His method of scientific inquiry, based on careful experimentation and precise recording, became a model for future generations of researchers. The accuracy and thoroughness with which Leeuwenhoek documented his observations were key to the acceptance of his findings, and they demonstrated the power of empirical research in advancing scientific knowledge. As Leeuwenhoek's discoveries began to attract attention, his reputation grew. In the years following his first observations, he continued to refine his microscopes and expand his investigations. He would send regular letters to the Royal Society, describing his findings and providing detailed drawings of the creatures he observed. His correspondence with scientists in England and elsewhere helped to establish him as a key figure in the scientific community, even though he was not formally educated in science and worked independently. His status as a self-taught observer set him apart from other scientists of his time, and his insistence on direct observation over abstract theorizing became one of the hallmarks of his approach. Leeuwenhoek's discoveries of microscopic life had profound implications for a wide range of fields. In medicine, the realization that disease could be caused by microorganisms, tiny creatures that were invisible to the naked eye, set the stage for the later development of germ theory. In biology, his observations of spermatozoa and microorganisms opened up new avenues of research into reproduction and the study of living organisms at the cellular level. Leeuwenhoek's work foreshadowed many of the scientific advancements that would follow, including the development of microbiology, pathology, and immunology. As Antony van Leeuwenhoek's discoveries began to challenge established scientific knowledge, his relationship with the Royal Society of London played a crucial role in advancing his reputation. The Royal Society, founded in 1660, was an esteemed scientific institution that served as a hub for intellectual exchange and the promotion of experimental science. Despite being a self-taught lens maker and microscopist, Leeuwenhoek managed to connect with this influential body, and it was through his correspondence with them that many of his most groundbreaking observations were shared with the wider world. The letters he sent to the Royal Society not only documented his discoveries, but also helped establish Leeuwenhoek as one of the most important figures in the scientific community of his time. In 1673, Leeuwenhoek sent his first letter to the Royal Society, marking the beginning of a fruitful correspondence that would span decades. The letter, directed to the Society's secretary, Henry Oldenburg, described the remarkable observations Leeuwenhoek had made with his microscopes. He detailed his discoveries of animalcules in pond water, microscopic organisms in saliva, and even tiny creatures found in the dust of household materials. These descriptions were accompanied by his intricate drawings, which showcased the extraordinary precision with which he had examined his specimens. This first letter laid the groundwork for Leeuwenhoek's future contributions to science, and it piqued the interest of the Royal Society's members, who were eager to learn more about this self-taught genius. Although Leeuwenhoek was not formally trained in science, his letters to the Royal Society were incredibly detailed and demonstrated a clear understanding of the scientific method. His ability to observe, document, and analyze the world around him was evident in every letter he sent. He wrote with such clarity that even those who had doubts about his abilities were compelled to take notice. His letters provided a wealth of information, not only about his discoveries, but also about the techniques he used to make them. For the first time, scientists in England were able to learn about the microscopic world, as Leeuwenhoek shared insights into his process for crafting lenses, using microscopes, and interpreting his findings. One of the most significant aspects of Leeuwenhoek's correspondence with the Royal Society was his dedication to sharing his observations in a systematic manner. In each letter, he carefully described the creatures he had seen, often in great detail. He described the movement of microorganisms, the structure of sperm cells, and the intricate features of insect anatomy. His precise descriptions left little to the imagination, and his drawings, which were often the result of hours of observation, brought his discoveries to life. Leeuwenhoek's ability to communicate his findings with such clarity helped the Royal Society recognize the significance of his work, even though he was not physically present to demonstrate his experiments. Despite the technical nature of his letters, Leeuwenhoek's correspondence was not just about science. It also revealed his personality. His letters were often written in a tone of quiet humility, as he was well aware of the limitations of his own knowledge. He expressed a deep respect for the members of the Royal Society and seemed eager to share his discoveries with those who had the intellectual capacity to understand their importance. Though his writing was respectful, it was also infused with a sense of wonder and excitement, reflecting the enthusiasm with which he approached his work. It was clear that Leeuwenhoek did not seek fame or recognition, but rather wished only to share his passion for discovery with others. The Royal Society's response to Leeuwenhoek's letters was a mixture of admiration and skepticism. Many of the Society's members were initially skeptical of the idea that such small, previously unseen organisms could exist. The concept of microscopic life was so far removed from accepted knowledge that it was difficult for some scientists to believe it without direct proof. However, Leeuwenhoek's persistence and the accuracy of his observations gradually won over the members of the Society. Over time, his letters were published in the Philosophical Transactions of the Royal Society, the prestigious journal that helped disseminate scientific discoveries throughout Europe. The publication of Leeuwenhoek's findings ensured that his work would reach a broad audience and cement his place in scientific history. Although Leeuwenhoek's discoveries were groundbreaking, he did not always receive the recognition he deserved. His work was often overshadowed by the more established scientists of his time, many of whom were highly skeptical of his claims. For example, Robert Hooke, one of the leading figures in the Royal Society, was initially cautious in his acceptance of Leeuwenhoek's discoveries. Hooke was a proponent of compound microscopes, and he doubted the effectiveness of the single-lens microscopes that Leeuwenhoek had used to make his observations. Despite these doubts, Hooke was ultimately forced to acknowledge Leeuwenhoek's skill and the accuracy of his findings, particularly after more scientists began to replicate his observations. In contrast to Hooke's initial skepticism, many of the Royal Society's members were more open to Leeuwenhoek's discoveries. As more letters were sent and more of Leeuwenhoek's detailed drawings were published, the scientific community began to appreciate the significance of his work. His contributions to the understanding of the microscopic world were unparalleled, and many scientists came to recognize him as a pioneering figure in the study of biology. In 1680, Leeuwenhoek was elected as a Fellow of the Royal Society, an acknowledgement of the impact his discoveries had on the field of science. Though he never became a full member of the Society, his election marked a milestone in his career and solidified his place in the annals of scientific history. Despite his newfound recognition, Leeuwenhoek remained an outsider in many respects. He was not part of the academic elite, and he continued to live a modest life in Delft, focusing primarily on his business and personal studies. His work with the Royal Society did not lead to the kind of fame or wealth that some of his contemporaries enjoyed, but it did bring him the respect of the scientific community. His relationship with the Society allowed him to continue his work without the constraints of formal education or institutional affiliation. This independence, coupled with his passion for discovery, made him one of the most innovative scientists of his time. Leeuwenhoek's letters to the Royal Society also had a lasting impact on the way science was communicated. His correspondence demonstrated the importance of direct observation and empirical evidence in the scientific process. By providing detailed descriptions and drawings, he showed that science could be pursued through careful and systematic investigation rather than relying on abstract theories alone. His letters became an invaluable resource for future scientists, many of whom would adopt similar methods in their own research. In this way, Leeuwenhoek's influence on the development of modern scientific practices extended far beyond his own discoveries. Antony van Leeuwenhoek's groundbreaking observations with the microscope did more than simply expand the frontiers of biology. They revolutionized the way science understood the living world. His discoveries of microorganisms, including bacteria, protozoa, and sperm cells, altered the trajectory of both biological science and medicine, paving the way for future research in microbiology, immunology, and pathology. The impact of his work was profound, though was not immediately recognized in its full scope. It would take time for the scientific community to fully appreciate the significance of Leeuwenhoek's findings and for his observations to be woven into the broader understanding of health, disease, and the nature of life itself. Before Leeuwenhoek's work, the concept of microorganisms was unknown, and the dominant belief in medicine and biology was that life forms could be spontaneously generated from non-living matter, a theory known as abiogenesis. The idea that diseases might be caused by invisible organisms or that life existed on a microscopic scale was unimaginable to many. Leeuwenhoek's detailed observations of animalcules in pond water, as well as his discovery of microorganisms in the human body, shattered these outdated ideas. By making the invisible visible, he brought to light a hidden world teeming with life, which not only challenged existing scientific theories, but also changed the way people thought about the natural world. In his letters to the Royal Society, Leeuwenhoek described his observations of various microorganisms, offering detailed descriptions of their shapes, sizes, and movements. He noted the diversity of these organisms, from the small, simple bacteria to the more complex protozoa, which exhibited various forms of locomotion. Leeuwenhoek's observations suggested that life could take many forms, even in environments previously thought to be lifeless. He demonstrated that microorganisms were not only part of the natural world, but were omnipresent, found in water, soil, air, and the human body. This was a revelation that altered the course of medical thought and laid the groundwork for the development of germ theory centuries later. Leeuwenhoek's discoveries were particularly influential in the realm of medicine, although their full implications would not be realized until much later. At the time, most medical practitioners relied on the humoral theory, which posited that illness resulted from an imbalance in the body's four humors: blood, phlegm, black bile, and yellow bile. Infectious diseases were often attributed to environmental factors or imbalances in these humors, but the idea of pathogens or microorganisms causing disease was not yet accepted. Leeuwenhoek's discovery of microorganisms in bodily fluids, such as saliva, feces, and semen, however, hinted at a connection between microscopic organisms and health. Although Leeuwenhoek did not make the connection between microorganisms and disease, his observations provided a foundation for future medical advances. His meticulous documentation of the microscopic world would influence the work of scientists and physicians who would come after him, including Louis Pasteur and Robert Koch. These later figures would build on Leeuwenhoek's work to develop the germ theory of disease, which posits that many diseases are caused by microorganisms. This shift in understanding would eventually lead to the development of vaccines, antiseptics, and antibiotics, revolutionizing medical practice and significantly reducing mortality rates from infectious diseases. In addition to his observations of disease-causing microorganisms, Leeuwenhoek's discovery of spermatozoa was a key turning point in the history of biology and medicine. His study of human semen in the 1670s marked the first time that sperm cells were observed under a microscope. Leeuwenhoek's detailed description of the motion of spermatozoa, which he observed swimming actively, provided the first direct evidence of the role of sperm in reproduction. Although the full implications of this discovery would not be understood until later, Leeuwenhoek's work laid the groundwork for the study of human fertilization and the science of genetics. Leeuwenhoek's focus on reproduction and the study of human bodily fluids also paved the way for future advancements in embryology and reproductive medicine. His discovery of sperm cells led to further research on human fertilization, and his observations of blood and lymph fluids opened new avenues for understanding the circulatory system. Leeuwenhoek's contributions to anatomy and physiology though indirect, had a lasting impact on the study of human biology. His work made it possible for future scientists to better understand the mechanisms of life at a cellular and microscopic level. In addition to his medical impact, Leeuwenhoek's discoveries also revolutionized the field of microbiology. Before his work, the world of microorganisms had been a complete mystery. While other scientists, such as Robert Hooke, had contributed to the development of the microscope, it was Leeuwenhoek's work that brought the microscopic world into sharp focus. His ability to construct high-quality single-lens microscopes allowed him to achieve magnifications that were far superior to those of his contemporaries. This gave him a unique ability to observe and describe the microscopic organisms that other scientists could not see. Although Leeuwenhoek did not possess the scientific knowledge or training of other prominent scientists of his time, his work set the stage for the later development of microbiology as a field of study. His emphasis on direct observation and empirical evidence became a cornerstone of the scientific method, and his meticulous record-keeping and drawings helped establish the importance of accurate documentation in scientific research. Leeuwenhoek's contributions to microbiology were pivotal in the establishment of the discipline, and his work influenced generations of scientists who would continue to explore the microscopic world he had revealed. Despite the enormous impact of Leeuwenhoek's discoveries, his work was not immediately recognized in its full context. At the time, his observations were seen as curiosities, and the significance of his findings was not fully appreciated by his contemporaries. While the members of the Royal Society recognized his talents and published his letters, the broader scientific community remained skeptical of his conclusions. It would take more than a century before Leeuwenhoek's work was fully incorporated into the developing theories of disease and biology. However, as time passed, his discoveries became more and more significant, and his legacy grew. Antony van Leeuwenhoek's discoveries with a microscope had a far-reaching influence, not only during his time, but also in the centuries that followed. His unparalleled ability to observe the microscopic world led to the creation of entire fields of study, including microbiology, pathology, and even aspects of immunology. While his work was groundbreaking, its true impact was not fully recognized until after his death. Over time, Leeuwenhoek's legacy has shaped the trajectory of modern science in ways that can still be seen today, both in laboratory research and in practical applications such as medical treatment, hygiene, and even biotechnology. The first and most significant aspect of Leeuwenhoek's legacy is his contribution to the field of microbiology. Before his work, the concept of microorganisms, be it bacteria, protozoa, or fungi, was completely foreign to science. Leeuwenhoek's detailed observations revealed a hidden world of life teeming in water, soil, and even in the human body. These discoveries challenged the prevailing scientific notions of the time and led to a rethinking of the basic principles of biology. While others, such as Robert Hooke, had contributed to the development of microscopes, it was Leeuwenhoek who demonstrated the full potential of the instrument. His lenses allowed him to make observations that revealed the intricate and varied forms of life on a microscopic scale, some of which were previously inconceivable. Leeuwenhoek's contributions laid the foundation for microbiology as a scientific discipline. It was his work that provided the early documentation necessary for the establishment of germ theory, which would later be formalized by figures like Louis Pasteur and Robert Koch. Pasteur's work on the relationship between microorganisms and disease and Koch's development of the germ theory of disease were directly influenced by the groundwork laid by Leeuwenhoek. His discovery of microorganisms in human saliva, feces, and semen foreshadowed the later recognition of the role that bacteria and viruses play in human health and disease. Leeuwenhoek's detailed letters and drawings proved invaluable as they provided the first visual evidence of life forms that were previously unseen, inspiring future generations of scientists to investigate the microscopic world in greater depth. Another area where Leeuwenhoek's legacy endures is in the study of human biology. His observations of human bodily fluids and their associated microorganisms paved the way for further exploration into human anatomy and physiology. His detailed descriptions of spermatozoa led to the eventual understanding of human reproduction, which would become a cornerstone of embryology and genetics. By examining the tiny creatures found in bodily fluids, Leeuwenhoek also opened the door for later investigations into blood circulation and immune responses. His work, though rudimentary by today's standards, was a key step in the quest to understand the inner workings of the human body, laying the groundwork for the study of cells, tissues, and organs. Moreover, Leeuwenhoek's emphasis on direct observation and empirical evidence helped establish the standards for scientific methodology. His insistence on documenting what he saw, often with precision drawings, influenced the way that future scientists approached research. In an era when many scientific theories were based on speculation or philosophical principles, Leeuwenhoek's approach was revolutionary in that it emphasized seeing as a prerequisite for understanding. His use of detailed, systematic observation became a model for future generations of researchers, particularly those in the fields of microbiology and pathology. The importance of rigorous data collection and verification that Leeuwenhoek exemplified continues to be a cornerstone of scientific inquiry to this day. In the broader scientific community, Leeuwenhoek's work was part of a shift from a theoretical, often philosophical, understanding of nature to a more empirical, evidence-based approach. His discoveries encouraged scientists to ask questions that were rooted in observation rather than assumption. The microscopic world was once thought to be insignificant or non-existent, but Leeuwenhoek's careful investigation demonstrated that there were entire ecosystems of life forms that could not be seen with the naked eye. This changed the way that future generations of scientists viewed the natural world. It was not only what was visible that mattered, but what could be discovered with the right tools and methods. Leeuwenhoek's work also spurred a new wave of innovation in the development of microscopes. His single-lens microscopes were advanced for their time, but they also served as a catalyst for future developments in optical technology. Though Leeuwenhoek did not invent the microscope itself, he was instrumental in refining its design. His ability to create lenses that provided such high magnification made it possible for other scientists to improve upon his designs. The use of microscopes became more widespread, and improvements in lens making eventually led to the compound microscope, which could magnify objects much more powerfully than Leeuwenhoek's single-lens models. Today, microscopes are indispensable tools in countless fields, from biology and medicine to materials science and environmental studies. In medicine, the long-term impact of Leeuwenhoek's discoveries is particularly pronounced. The awareness of microorganisms as agents of disease that his work helped inspire paved the way for advances in hygiene, sterilization, and infection control. His work eventually contributed to the development of antiseptics and vaccines, which revolutionized public health. Though Leeuwenhoek did not directly make the link between microorganisms and disease, his discoveries made it possible for later scientists to form that connection. This paved the way for the prevention and treatment of infectious diseases, which have saved millions of lives over the past century. Leeuwenhoek's influence also reaches into modern biotechnology. The field of genetic engineering, which involves manipulating the genetic material of organisms to produce useful products, draws on many of the foundational concepts that Leeuwenhoek's work helped establish. His detailed observations of microorganisms made it possible for future scientists to understand the role of bacteria and other microbes in biological processes. Today, bacteria are used in a variety of biotechnological applications, including the production of insulin, antibiotics, and genetically modified crops. Though Leeuwenhoek could not have foreseen these developments, his work contributed indirectly to the research that would eventually make biotechnology a reality. Furthermore, Leeuwenhoek's legacy lives on in the growing field of environmental microbiology. The study of microorganisms in natural environments such as soil, water, and air has its roots in his work. Modern environmental scientists study the ways in which microbes interact with their surroundings, contributing to processes like nutrient cycling and the breakdown of pollutants. Leeuwenhoek's discovery of microorganisms in water and soil helped plant the seeds for this interdisciplinary field, which is now critical for understanding environmental issues such as pollution, climate change, and biodiversity conservation. One of the most enduring aspects of Leeuwenhoek's legacy is the way in which it bridged the gap between theory and empirical science. His emphasis on the observable world, his insistence on using empirical evidence to validate theories, and his documentation of findings in written form set a new standard for scientific communication. His detailed letters to the Royal Society, filled with precise observations and intricate drawings, continue to be a model of scientific documentation. In a world where information spreads more quickly than ever, the importance of clear and accurate reporting remains as relevant as it was in Leeuwenhoek's time. While Antony van Leeuwenhoek is celebrated today as one of the most groundbreaking figures in the history of science, his personal life remains less well known. Born into modest circumstances in the Dutch city of Delft in 1632, Leeuwenhoek was the son of a cloth merchant and grew up in a world far removed from the scientific community that would later elevate him to international fame. Despite his extraordinary contributions to science, Leeuwenhoek led a relatively quiet and private life, much of which was devoted to family, business, and his personal curiosity. His work as a scientist was, in many ways, a reflection of his insatiable desire to understand the world around him. But this drive coexisted with the responsibilities and interests of a man deeply embedded in his local community. Leeuwenhoek's early life was characterized by a combination of business and education. His formal education was minimal, but he did attend the local school in Delft where he received the basic literacy and numeracy skills that would serve him throughout his life. Although he was not trained in the sciences, he had a natural curiosity and an innate understanding of how things worked. After leaving school, Leeuwenhoek worked for a time as an assistant to a merchant, but his life would take a different turn when he started his own business. He eventually became a successful draper, and his work in the textile trade allowed him to support himself and his family while pursuing his personal interests in microscopy. Despite his professional success, Leeuwenhoek's passion for microscopes and lenses would come to define much of his life. Although he was primarily known for his work as a lens maker, he was also deeply involved in the maintenance and repair of scientific instruments. His work on microscopes began as a hobby, and it quickly consumed him. Leeuwenhoek would spend countless hours constructing lenses, perfecting his microscopes, and making observations of the natural world. His ability to make incredibly powerful microscopes with a single lens was a skill that set him apart from his contemporaries, and it was this craft that would allow him to uncover the microscopic world that few others could see. Leeuwenhoek's personal life also involved his role as a family man. He married twice in his lifetime, and both unions played important roles in shaping his character and his career. His first marriage was to a woman named Barbara de Mulder, with whom he had several children. Though little is known about his first wife, it is clear that she was an important figure in his early life. Tragically, Barbara passed away in 1666, leaving Leeuwenhoek to care for their children. In 1671, Leeuwenhoek married his second wife, Cornelia Swalmius, and together they had one daughter. This second marriage seemed to provide him with a sense of stability, and Cornelia supported him in his scientific endeavors, allowing him the freedom to focus on his work. Although Leeuwenhoek was deeply dedicated to his family, his life was not without its challenges. As his scientific pursuits gained recognition, he often found himself embroiled in disputes with colleagues and fellow scientists. While he was well respected for his discoveries, his unorthodox methods and lack of formal education made him a somewhat controversial figure in the scientific community. At times, he was criticized for not sharing his knowledge and findings more freely, and his secretive nature added to the mystique that surrounded him. Leeuwenhoek was a solitary figure, preferring to work alone rather than collaborate with others, which was not always well received by those in the Royal Society or the broader scientific community. His reluctance to share his techniques for making lenses, for example, created tension with other scientists who were eager to learn from him. Beyond his scientific endeavors, Leeuwenhoek was a man of considerable curiosity and intellectual vigor. He kept an extensive journal of his observations and experiments, and his meticulous records offer a window into his methodical approach to science. His diary entries are filled with details of the specimens he observed and the lenses he constructed, as well as reflections on his experiments and the difficulties he encountered. Leeuwenhoek's writing reveals a man who was driven by an insatiable desire to understand the world, yet who also grappled with moments of doubt and uncertainty. His humility, as evident in his correspondence with the Royal Society, reflects his awareness of the limits of his own knowledge. Though he was an extraordinary observer, he often seemed unsure about how to interpret his findings, a trait that humanized him in the eyes of his peers. Leeuwenhoek's relationship with the scientific community, particularly the Royal Society of London, was complex. On one hand, his discoveries were groundbreaking, and his meticulous records were admired by many. On the other hand, his reluctance to share his knowledge freely, his solitary nature, and his refusal to adhere to traditional scientific conventions made him a somewhat enigmatic figure. While the Royal Society, in particular, recognized the importance of his work and published his letters, they were often frustrated by his secretive approach to science. Leeuwenhoek's refusal to divulge the full details of his techniques for making his microscopes, in particular, was a source of contention. Yet, despite these tensions, the Royal Society continued to value his contributions, and Leeuwenhoek's correspondence with them helped establish him as a figure of great scientific importance. In many ways, Leeuwenhoek's personal philosophy was shaped by the same curiosity that guided his scientific work. He sought knowledge for the sake of knowledge, not for fame or financial gain. His love of discovery was genuine, and he often expressed astonishment at the things he observed through his microscopes. This wonder is evident in his letters, where he describes his excitement upon encountering new microorganisms or discovering a previously unknown aspect of the natural world. It is clear that Leeuwenhoek saw his work not as a means to an end, but as a continuous journey of exploration and understanding. His desire to uncover the mysteries of life was, in many ways, an extension of his own personal quest for meaning and insight. Though Leeuwenhoek was a man of considerable intellect and curiosity, he was not immune to the common struggles and challenges that every person faces. He suffered personal losses, such as the death of his first wife and the challenges of raising children on his own. He also experienced professional frustrations, particularly as his work was not always accepted by the broader scientific community. Despite these challenges, Leeuwenhoek persisted in his efforts, driven by a passion that would ultimately change the course of scientific history. His resilience in the face of adversity is one of the key aspects of his character that contributed to his success as a scientist. Ultimately, the life of Antony van Leeuwenhoek reveals a man who, though not formally trained in science, had an extraordinary ability to observe and understand the natural world. His personal and professional life were intertwined with his passion for discovery, and his contributions to science were fueled by a deep curiosity and a relentless drive to explore the unknown. Leeuwenhoek's legacy extends far beyond his scientific work. It is a testament to the power of individual curiosity and determination in the face of doubt and adversity. His life shows that great achievements in science can come from unexpected places and from those who are willing to look beyond the surface to uncover the hidden truths of the world around them. Antony van Leeuwenhoek's remarkable scientific contributions were not born from a formal academic training or a prestigious position within the scientific community. Instead, they emerged from a mind of a self-taught observer who navigated significant personal, social, and professional obstacles throughout his life. Despite his groundbreaking discoveries in microbiology, Leeuwenhoek faced numerous challenges, both in the development of his microscopes and in gaining recognition for his work. His journey as a scientist was not smooth or easy, and his story is one of resilience, perseverance, and overcoming the limitations placed on him by both his personal circumstances and the scientific landscape of his time. One of the primary challenges Leeuwenhoek faced was his lack of formal scientific education. Born into a modest family in Delft, Leeuwenhoek did not attend university or study under established scholars. His education was limited to the local schools in Delft, where he learned basic literacy and numeracy. He had no advanced training in biology, physics, or any of the fields that would later inform his scientific investigations. Yet, despite this, Leeuwenhoek's curiosity and ingenuity allowed him to craft his own path. He taught himself the principles of lens making, built his own microscopes, and relied on his keen observational skills to make discoveries that would astound the world. His lack of formal credentials meant that, for much of his career, he was not regarded as a legitimate scientist by many of his contemporaries. In addition to the lack of academic training, Leeuwenhoek faced another significant obstacle: the skepticism of the scientific community. Although he was a member of the prestigious Royal Society of London, his contributions were often met with doubt and resistance. Many members of the scientific establishment struggled to accept that a man with no formal education could make such revolutionary discoveries. His colleagues were particularly skeptical of his methods. Leeuwenhoek's reliance on his self-made microscopes, combined with his somewhat unorthodox way of presenting his findings, led some to question the validity of his observations. Furthermore, his refusal to share his methods and techniques for lens making only deepened the suspicion surrounding his work. This reluctance to collaborate with others, common in Leeuwenhoek's work, was seen as a significant barrier to wider acceptance within the scientific community. Perhaps one of the most unique challenges Leeuwenhoek faced was the practical difficulty of constructing his microscopes. The process of lens making was an arduous and delicate task. Leeuwenhoek did not have access to the advanced equipment that would later be developed in the field of optics, and he had to rely on his own ingenuity to grind and polish the lenses by hand. The skill required to produce high-quality lenses capable of achieving the magnifications necessary to observe microorganisms was extraordinary. Moreover, Leeuwenhoek's microscopes were single-lens models, which were less advanced than the compound microscopes that were eventually developed by other scientists. Yet, despite these limitations, Leeuwenhoek was able to create microscopes that were vastly superior to anything available at the time. The precision and craftsmanship that went into the making of these lenses were crucial to his success, but it also meant that his work was slow and painstaking. Another challenge Leeuwenhoek faced was the isolation that came with being a self-taught scientist. In an era when scientific inquiry was often pursued within well-established academic circles, Leeuwenhoek worked largely alone. His decision to keep his methods secret, particularly his techniques for making microscopes, further alienated him from potential collaborators. While some scientists in the Royal Society admired his work and published his letters, Leeuwenhoek's lack of openness about his techniques left many frustrated. He was not part of the larger academic network that exchanged ideas and shared knowledge. Instead, he was an isolated figure, driven by his own passion for discovery. While this autonomy allowed him to maintain control over his work, it also meant that he had little support or guidance from other scientists, which could have helped to refine and validate his findings sooner. Leeuwenhoek's relationship with the Royal Society, though initially one of admiration, was complicated by his unwillingness to conform to the expectations of scientific collaboration. While he regularly corresponded with the Society, sending them letters that described his discoveries in minute detail, he often withheld the specifics of his methods. This created tension, as many members of the Society wanted to know how Leeuwenhoek achieved such extraordinary magnifications with his microscopes. His refusal to reveal these secrets fueled doubts about the reliability of his work. Despite this, the Royal Society recognized the value of his observations and eventually published many of his letters, which contributed to his recognition as one of the foremost scientists of his time. Still, it is clear that Leeuwenhoek's reluctance to fully engage with his peers hindered his ability to gain wider acceptance within the scientific community. In addition to the skepticism he faced from his peers, Leeuwenhoek also grappled with the challenge of communicating his discoveries to a public that was largely unprepared to comprehend the significance of his work. The world of microorganisms he uncovered was invisible to the naked eye, and many of his discoveries were so foreign to the scientific knowledge of the time that they seemed almost fantastical. For instance, his descriptions of the animalcules found in water were met with skepticism because they seemed to contradict existing theories about life. The concept of such tiny, complex organisms existing in nature was beyond the comprehension of many, and without the advanced knowledge of biology and medicine that we have today, it was difficult for people to grasp the implications of his work. Leeuwenhoek's letters, while detailed, were often filled with descriptions that sounded more like curiosity than hard scientific evidence, and it took time for his findings to be understood and integrated into the broader scientific narrative. Despite these challenges, Leeuwenhoek's determination to pursue his research never wavered. He continued to make discoveries well into his later years, driven by an unyielding passion for the natural world. Even as he grew older and faced the physical limitations that come with age, Leeuwenhoek remained committed to his craft. His work in lens making and microscopy continued, and he continued to publish his findings for the Royal Society. His perseverance in the face of adversity, both personal and professional, allowed him to achieve an unparalleled legacy in the world of science. The challenges Leeuwenhoek faced in his scientific career were not limited to the obstacles imposed by his lack of formal training or his relationships with other scientists. He also faced personal hardships, including the loss of his first wife, Barbara, and the responsibilities of raising children on his own. These personal challenges no doubt affected his work, yet they also shaped his character and strengthened his resolve. Leeuwenhoek's ability to continue his research despite personal tragedy and professional obstacles speaks to his extraordinary dedication to science and his relentless pursuit of knowledge. Ultimately, the struggles Leeuwenhoek encountered in his life only served to amplify the significance of his achievements. His lack of formal training, isolation from academic circles, and reluctance to share his techniques might have limited his immediate recognition, but they did not prevent him from uncovering the microscopic world that had eluded others for centuries. Despite the challenges, he remained committed to his passion and was able to make discoveries that have had a lasting impact on science and medicine. Leeuwenhoek's story is one of overcoming adversity through perseverance, resourcefulness, and an unwavering commitment to scientific inquiry. His life serves as a reminder that great achievements often come from those who are willing to face and conquer the challenges that stand in their way. Antony van Leeuwenhoek's legacy, though initially overlooked, has grown into one of the most profound in the history of science. While he may not have been a member of the academic elite during his lifetime, his discoveries laid the foundation for the entire field of microbiology and revolutionized our understanding of the natural world. His meticulous observations through his powerful microscopes and his pioneering work in studying microorganisms have earned him the title of the Father of Microbiology. Today, Leeuwenhoek's name is forever linked with the birth of a new era in scientific thought, one in which the microscopic world is revealed and the invisible forces of nature are brought to light. The most immediate and enduring impact of Leeuwenhoek's work is his contribution to the discovery of microorganisms. By observing and documenting life forms too small to be seen with the naked eye, he essentially opened up a new dimension of biology. His descriptions of bacteria, protozoa, and spermatozoa, often referred to as animalcules, were some of the first insights into a world that had previously remained hidden. Leeuwenhoek's early studies demonstrated that life was far more complex than anyone had imagined, with a vast and diverse range of organisms existing beyond the limits of human vision. These discoveries reshaped the way people viewed life on Earth and set the stage for later developments in biology, including the understanding of diseases, the development of antibiotics, and the eventual formulation of germ theory. Leeuwenhoek's work on microorganisms provided evidence for the existence of life forms at a microscopic level, but his influence extended far beyond the simple identification of new species. His work fundamentally challenged the scientific worldview of the time. The prevailing theories of biology and life were deeply rooted in the traditions of Aristotle and other ancient scholars, who believed that life existed in a fixed and hierarchical manner. Leeuwenhoek's discovery of minute organisms that were both abundant and diverse disrupted these long-held beliefs. He showed that life was not simply a matter of large, visible organisms, but was composed of many invisible forms that played crucial roles in the functioning of ecosystems. His discoveries contributed to the eventual acceptance of a dynamic, evolving view of life that would be essential to modern biology. One of the key aspects of Leeuwenhoek's legacy is his meticulousness as an observer. He was not content to simply see new things. He sought to understand them in intricate detail. His letters to the Royal Society, filled with exacting descriptions of his observations, reveal a man who approached science with the precision of an artist. Leeuwenhoek was an extraordinary detail-oriented observer, not only documenting the appearance of microorganisms, but also observing their behavior and movement. His notes show that he carefully studied how these creatures moved, interacted, and behaved in their environments. This focus on the living and dynamic nature of microorganisms was groundbreaking, as most earlier studies of natural history had emphasized static classification. Leeuwenhoek's attention to behavior was crucial for the development of later concepts in microbiology and ecology. The advancement of microscopy itself owes much to Leeuwenhoek's innovation. His microscopes were not the first, but they were undoubtedly the most advanced of his time. The simplicity and effectiveness of his single-lens microscopes gave them remarkable magnification power, enabling him to peer into the microscopic world with unprecedented clarity. Leeuwenhoek's lenses were so finely crafted that they could magnify objects up to 300 times, allowing him to discover details of the natural world that were previously unimaginable. His expertise in lens-making not only advanced the technology of microscopy, but also set the stage for future innovations in optical instruments. The techniques Leeuwenhoek developed in creating these lenses were highly secretive, and although he did not share them widely during his lifetime, they became a touchstone for later scientists who sought to improve the design of microscopes. Leeuwenhoek's contributions also had far-reaching implications for medicine. By observing the microorganisms in saliva, blood, and other bodily fluids, he unknowingly laid the groundwork for later discoveries related to infectious diseases. Although the germ theory of disease would not be fully established until the 19th century, Leeuwenhoek's observations revealed a hidden world that would eventually be linked to the causes of various illnesses. His work pointed toward the idea that tiny, invisible organisms could be responsible for disease, a concept that was revolutionary at the time. In this way, Leeuwenhoek's discoveries opened up new avenues of research in the study of pathogens and the development of medical treatments, which would eventually lead to the birth of microbiology as a field dedicated to understanding the role of microorganisms in health and disease. Perhaps one of the most enduring elements of Leeuwenhoek's legacy is the model of scientific inquiry he exemplified. Although his methods were unconventional and at times controversial, his passion for exploration and his relentless pursuit of knowledge served as an inspiration for future generations of scientists. Leeuwenhoek worked largely outside the established academic structures of his time, choosing instead to follow his own curiosity and observe the world on his terms. His approach was grounded in practical observation and empirical evidence, emphasizing the importance of seeing the world for oneself rather than relying on preconceived notions. This independent spirit, combined with his ability to make groundbreaking discoveries from a place of self-reliance, made Leeuwenhoek a role model for the scientific method as we know it today. Leeuwenhoek's impact on the scientific community was also profound because he was one of the first to recognize the importance of documenting his findings in a systematic way. His correspondence with the Royal Society was key in disseminating his discoveries to a wider audience, and the letters he sent were carefully crafted records of his observations. These letters not only communicated new scientific insights, but also reflected the careful thought and precision that went into his work. By contributing to a growing body of knowledge through written communication, Leeuwenhoek helped lay the foundations for the modern scientific literature that we rely on today. Over time, the importance of Leeuwenhoek's work was gradually recognized, especially after the development of more advanced microscopes in the 19th century. His name became synonymous with the study of microbiology, and his contributions were incorporated into the broader narrative of scientific progress. Today, Leeuwenhoek's legacy is celebrated in countless ways. His work is foundational to fields like bacteriology, virology, and parasitology, which continue to have profound effects on human health and disease prevention. Many institutions dedicated to scientific research, including microbiological societies, museums, and academic programs, honor Leeuwenhoek's role in shaping the trajectory of life sciences. Moreover, Leeuwenhoek's approach to scientific discovery continues to resonate in modern scientific practices. His willingness to ask questions that challenge the status quo, his emphasis on direct observation, and his ability to observe phenomena that others had missed have made him an enduring symbol of scientific curiosity and innovation. His life and work demonstrate that even without formal training or recognition, a deep passion for discovery and a commitment to rigorous observation can lead to groundbreaking insights that change the course of human knowledge. In conclusion, Antonie van Leeuwenhoek's legacy is one of profound influence that continues to shape the fields of biology, medicine, and microscopy. His pioneering work on microorganisms, his contributions to the development of the microscope, and his dedication to careful observation have made him a figure of immense importance in the history of science. Today, he is remembered not only as the father of microbiology, but also as a symbol of the power of curiosity and the importance of scientific inquiry. The microscopic world that Leeuwenhoek first uncovered is now an integral part of our understanding of life itself, and his discoveries remain a testament to the lasting impact one person can have on the course of human knowledge.